Anyway, it's a pleasure for me to join you today. Good afternoon. I'm going to attempt to share my screen. And of all those things that Leo said that I was interested in, what's really captivated me now is in the last 5 years, I would say, is development of new tools for single cell analysis of microbial communities. Let me see if I can get this up in presentation mode and get a pointer. Here we go. Okay, so you see that my titles talk, my talk title, excuse me. And I won't dwell on that. And I basically, oh, I can't even read this, I'm sorry. Oh, there we go. Okay. Title is Exploring the Microcosmos. And why do we want to do that? Well, by now in this semester, you should all be familiar with the biological carbon pump and a bit about the microbial loop. And layered on the basic food web and the biological carbon pump, there's a whole array of microbial interactions that are at play that we know so little about. Most of the measurements we make are at the community level, and they don't allow us to really resolve some of these interactions. And your Professor Foster has been pioneering in this area as well, looking at symbioses. Am I advancing? What's going? There we go. Okay, so what are some of our motivations for studying the microcosmos? Well, in oceanography, most of our traditional methods for biogeochemical measurements and exchanges only allow us to look at net transformations, and they're at scales that aren't really relevant to microorganisms. The current molecular tools like ribosomal RNA gene libraries, metaomics, DNA, RNA, SIP, et cetera, they're analyzing largely community properties where many of the interactions we're interested in are at the level of individuals and populations. Biogeochemical reactions are occurring over in microenvironments. They involve cell-to-cell interactions. And many of the techniques that we've used traditionally are blind to these interactions. So many of us have, in the last decade, I would say, really paid a lot more attention to what I'm calling microspatial seascape ecology, and looking at individual plankters, both out in the field and in culture. And these are plankters whose universe doesn't extend beyond 1 cubic centimeter. There's a lovely paper by Roman Stocker that talks about that 1 cubic centimeter, that spatial scale that most microorganisms, that's all they know, that's their microcosmos. But we barely explored this in the plankton. And as I'm fond of saying for quite some time, our holy grail as microbial ecologists is to be able to convincingly link biogeochemical functions, activities that we measure in bulk, to the identity of the key players. And being able to do this will allow us to make more authoritative inferences from these massive genomic databases that we are gathering. So current practices, to sum up, are largely assess inter and intrapopulation variability, poorly assess, in physiological responses. Cryptic exchanges are overlooked. Key players are often not recognized. And many of the techniques that we use don't really allow us to delve into the mechanistic underpinnings of ocean processes. So we're searching for tools that allow us to do that more effectively. Okay, respond. I'm not responding here. Oh, here we go. All right, so to continue on motivation, back when I started in microbial ecology, our perception of the plankton was more or less like this upper micrograph right here, and that's obviously out of date and naive. I'm having trouble controlling here we go. But we've come to realize that at small scales, the plankton is very heterogeneous. And this is a rather famous doodle from Farouk Azam. I think he did it on a napkin at a bar or restaurant, and it got published in Nature or Science. I can't remember where. Anyway, it probably looks a little bit more like that. So the physical and chemical composition of the seascape is really at microspatial scales that matter to microorganisms. It's very heterogeneous, much more so than we ever imagined, showing what 1 cubic centimeter may look like if you could really see it in those 3 dimensions. And it's, as you can see, there's a lot going on here. It's very, very heterogeneous. And bulk measurements that we make don't inform us about what's going on there. We have small spatial scale gradients in all kinds of nutrients and organic substrates that are caused by decomposition, cell lysis, etc. They can be very important in which organisms dominate and what their activities are. Our understanding of symbiosis, competition, cooperation, allelopathy, and parasitism amongst the microbes, our understanding has been impeded by the blunt instruments that we use. And these are just some examples of various interactions that have been examined at the micro scale. Okay, so what are the ways forward? Well, I'm sure you've heard time and again with many of the guest lecturers that cultivation independent approaches to link function and identity is really important. There are many practitioners of RNA, DNA, SIP, stable isotope probing, which I assume you've heard about so far at some point. A rather old technique that has come in and out of fashion is MARFISH, microautoradiography fluorescence, in situ hybridization, where we use radio tracers. I'm not going to go into the details of any of these. Flow cytometry has been very, very powerful technique. Coming into play now are microfluidics, which I really don't have time for, and I'm sure you've heard about SIP nanosims, given that Professor Foster is a user of that technology. Single cell amplified genomics has taught us a lot about how heterogeneous microbial populations are. We even have folks that do single viral genomics, one of whom I'm collaborating with these days. Another tool that we use, but I really won't discuss, is atomic force microscopy. I will show a few data or figures from AFM. But what I'm going to focus on now is SIP Raman fish, or the use of Raman microspectroscopy in particular. And these approaches that I've listed here really are a gateway to interrogating natural communities as well as cultivated model systems. Okay, what is Raman scattering? Well, it's not this. And in fact, Raman scattering is spelled R-A-M-A-N rather than E-N, kind you eat. Raman scattering is a form of vibrational spectroscopy, complementary to infrared spectroscopy. And it's based on scattering of monochromatic light, that is laser light. It was predicted back in 1923 that this effect existed, but it was proven experimentally by C.V. Raman in 1928. And it basically provides quantitative information on chemical bonds, and it does so non-destructively. and it gives what we would call chemical fingerprints. And for this discovery, Raman received the Nobel Prize in 1930, first Indian to ever receive a Nobel in science. So let me just show you briefly what Raman's all about. We shine laser light onto a target, a specimen, a molecule, a mineral, a cell, and 1 in a million or 1 in 10 million of those photons actually collides with an atom and causes it to vibrate. You can think of it as two atoms linked together with a spring, and the atoms are vibrating away and towards one another. The frequency with which they vibrate is characteristic of which two atoms are bound and the nature of the bond. And what we actually measure are called the Stokes shifted photons. So these are photons that have given up a little bit of energy to the chemical bond or the molecule that they've interacted with, and therefore have less energy and are at a longer wavelength. And the spectrophotometer collects those photons and produces what's shown down here, a characteristic spectrum. This is a very idealized spectrum that you might get from a mineral that doesn't look like that in a cell. There are all kinds of vibrational modes, which I've summarized in these figures here. There's bending, there's stretching, there's asymmetric stretching, symmetric stretching. In aromatic compounds, there's a ring breathing mode. When the photon hits them, it causes them to vibrate at a characteristic frequency. Okay. Using this technology, we can actually, we couple it to a microscope, and this is a published image of a bacterial culture and the spectrum from an individual bacterium. And we have peaks that are characteristic of various macromolecules within the cell. You can see them listed here. This is also a little bit idealized. It's often not as clean as this, but basically allows us to see all the important macromolecules in an individual cell. It is a weak phenomenon, so you need very sensitive instrumentation. It is, well, I've already mentioned this, it measures the frequency due to the energy transfer from the incident light to the chemical bond. And it allows us to see some subtle chemical differences. And in fact, it is sensitive to the isotopic signature, which we'll come to in a moment. Anyway, these peaks collectively tell us something about the identity of the target. What does the instrument look like? Well, there's
excuse me.
Yes.
So I just want to make sure I got this right. So Raman scattering is a way so we can see like what kind of macromolecules does the individual cell contain.
That's correct. And this technology is broadly applicable. It's used in material science and chemistry. So my application, of course, is microbiology, and that's why I'm talking to you today. But Raman is used for all kinds of chemical analyses, gases, minerals, and so on. Anyways, yes, but you've got the point. That's correct.
And can we like do a picture of it also?
Yes, and I will show you in upcoming slides where we get simultaneous photographs, either fluorescent or bright field, as well as chemical information.
Okay, great.
We will come to that. All right. So in 2014, I was fortunate enough to get a large major research instrumentation grant from our National Science Foundation that allowed me to build this facility. And we have a Renishaw Raman microspectral photometer. That's a mouthful, isn't it? There's the instrument right there. What you don't see behind the instrument back here are 3 different lasers. And so we can produce laser light actually at 4 different wavelengths with our setup. Under this dome right here is essentially a epifluorescent microscope. It's a Leica microscope. And the dome is to prevent laser light from being scattered around the room and endangering the user. But basically, it is built around a microscope. There's a camera up here, highly sensitive to get our images. And the unique feature of our setup is that on the same benchtop, we have an atomic force microscope. Here's the working stage of the atomic force microscope. And we are able to optically couple the Raman to the atomic force microscope, send laser light through this black tube, focus it through a objective lens. I'm circling it right here, and point that laser light at the tip of the AFM sampling tip, which is about 10 nanometers in diameter. So we can resolve things down to on the order of 20 to 30 nanometers on a surface using our atomic force microscope. That's well beyond what I'm going to be talking about today, though. A schematic of the system, I don't want to dwell on this, but basically we have the 4 different laser lines we can produce. The laser is all conducted internally. We never see the laser light. It goes through the spectrophotometer. It goes down through the microscope stage and samples the substrate from above. Prior to taking introducing laser light and getting the spectrum, we close the shutter and we can get a visual image. We can then photograph it and then change the position of the shutter here, and then we are able to get the spectrum. And as I said, the unique feature of our system is that we can redirect the laser light rather than to the microscope, the optical microscope. We can send it to the atomic force microscope and look at, get chemical information at a 10-fold higher spatial resolution. See smaller spots. Okay, but again, that's, well, I'll stop what I was saying because I am going to show you one bit of data, not from our lab, but published data. When you couple atomic force microscopy with Raman, you can actually get spectral information from an individual strand of DNA. This image you see here is a combed strand, double-stranded DNA molecule on the atomic force microscope stage. The laser did a transect across the DNA molecule, and here are the spectra that were obtained that give us the signature of the base pairs that were in this portion of the DNA. So it's a very powerful, high-resolution technique, but it is very challenging. This is an enhancement technique called tip-enhanced Raman scattering. And again, that's kind of beyond the scope of what I want to talk about today, but it is a new frontier. Okay, what I'm going to do now is talk, I'm calling them vignettes of 3 different projects or applications that we've used Raman for. And the first one, I sent you all the paper, or I sent it to Professor Foster, and she gave it to you, so you may already be familiar with this, so I won't dwell on it too long. But we've used stable isotopic probing combined with Raman to actually measure individual growth rates of single Synechococcus cyanobacterial cells in culture. The experimental design, just this perhaps is review for you guys since you read the paper, is that we set up a set of parallel incubations where we grew Synechococcus at different levels of C13. The fractional abundance, F media, symbolized as F media, is shown diagrammatically. Natural abundance, 11%, up to 54%, carbon-13 bicarbonate. Our instrument gives us epifluorescent micrographs, so this is an autofluorescent micrograph of the targets that we're going to analyze. And I only show this graph to ease your mind about, well, what is the effect of adding all that heavy isotope? Well, there's no apparent effect on growth. no matter how much of the C13 added, all the cultures are growing at the same rate. Okay, so we can use this technique because basically the level of labeling in a culture such as this, when the bicarbonate is the only carbon source, is predictable. If we were to assume that the carbon-13 was 100% of all the DIC that the cyanobacteria would assimilate, at time 0, they'd be 1% C13. After 1 generation, they'd be nearly 50%. After 2 generations, 74, and so on. So this is entirely predictable, and we can model that mathematically. And if we use different levels of labeling, you generate curves like this. And the point here is that over a sufficient number of generations, the cells will approach equilibrium with their media. So if it's 10% C13, we approach 10%, 20%, we approach 20%, etc. And it approaches, in a predictable fashion, equilibrium within 6 to 8 generations. So we're showing generations here rather than time. So in one example where we have natural abundant C13 and a lot of C13, 96%, we generate these spectra. Question? No? Okay. So this is a stacked set of spectra. The upper spectrum is from an individual cyanobacterium. It's a single spectrum that took 2 seconds to acquire from this field. So this is a bright field micrograph of all the targets, and all those little red dots are actually numbers. They're very, very small, but each one is a numbered target. This is the same field on the under epifluorescence illumination. And then we open the shutter for the laser, and we acquire in 2 seconds a spectrum from each one of those targets. Natural abundance, we see 3 major peaks that are attributable to carotenoids. And then the spectra that are underlying or below are from organisms, cultures that were incubated with heavy C13. This is after 5 days, 6 days, 8 days, 9 days. And what you notice in all 3 of these major peaks is there is a leftward shift in the frequency, or in this case, we use the units reciprocal centimeters, which is wave number rather than frequency. And it is a predictable shift. We call this a red shift because the lower frequency, that's a warmer color. It's moving towards the red part of the spectrum, and it is occurring in a very predictable fashion. What's going on in these spectral shifts? Well, go back to this diatomic model that I showed earlier. If you have isotopically light molecules, they will vibrate at 1 frequency. If we have an isotopal log that is C12, C13, it has an intermediate frequency. And if both atoms are C13, it has a slower frequency because it's heavier. And so it can't vibrate as fast with the same photon energy input. And that's being illustrated here. This is one of the spectral peaks from carotenoids under natural abundance, where 54% or 0.54 of the media is C13. And 3 days, we see a peak that is attributable just to the C12, C12, and one that is attributable to the C13, C12. Incubate longer, and we see that the relative peak areas and heights of these 2 peaks change, and we see the appearance of uniformly labeled carbon as well with the lowest frequency. So 1, 2, 3 are shown here. So that's the idea behind stable isotopic probing and Raman detection. That experiment that I showed earlier, where we had multiple concentrations of C13 and a time course, were summarized in these figures here. And basically what we're comparing is the isotopic abundance of the cells, or the fractional abundance in the cells, how much C13 per total carbon, 12N13, is there, and what is the effect on the wave number or frequency of those 3 Raman peaks. And you can see there is a very predictable shift. And each one of these dots is the average of 25 points, and the standard deviation is around them. So you see it's very, very sensitive to isotopic change. And we can detect about a 3%, changes as little as about 3%. We can't see natural abundances. The technique is not that sensitive, but for these enrichment experiments, it's no problem. We're not going to go through the math, but this is just to remind me that we can then use the Raman spectral data to derive, from the Raman spectral data, we can derive how isotopically heavy it is. And knowing how long it's been incubated, we can actually calculate the specific growth rate of single cells, each individual target. I'll remind you, data took 2 seconds to get for each cell. So the data throughput is pretty decent. And then you need, as validation, to compare it to an independent method. And that's what we illustrate here, where we've in parallel measured the growth rates by in vivo fluorescence. So we've gotten the population growth rate, and we're comparing the population growth rate with the individual growth rates. And there's a statistical technique that's used often in biomedical sciences called planned Altman plots, where you compare the difference between measurements that were made by 2 independent methods, Raman for single cell, in vivo fluorescence for population. You compare the difference to the average of those 2 measurements. And then you look at where the yellow area is the 95% confidence interval, and anything that falls within that, you're confident. You can confidently say that both techniques are measuring the same thing. 97% of our observations fall within those confidence intervals. Okay, probably spent more time than I meant to on that. The second project that I'm going to talk about is it's actually summarizing paper that we're just about to send off for publication. Thought it would be out last week, but many things have happened. Anyway, we're looking at viruses using Raman and atomic force microscopy. We're using a model system, and the host is the coccolithophore Emiliania huxleyi, which is a globally important coccolith. It forms these massive blooms in many portions of the ocean that you can see from outer space. This is the Cornwall coast off of the UK. That milky water is because of these calcifying coccoliths, and they're only about 8 microns or so in diameter. They're rather small eukaryotic microalgae. We have in culture a virus that attacks them and is responsible for terminating these massive blooms, or at least partially. We don't know to what extent, but we think it's very important in terminating these massive coccolith blooms. And this is an AFM image of the individual viruses that we've harvested from an infected culture, cleaned it up, and then deposited the viruses on a very flat surface, which is mica. Here's a close-up of an individual. It's EHV-163 virus. It is an enveloped virus, like many human viruses. It's kind of unusual in marine systems. to have enveloped viruses, but this one happens to be. This is the AFM image, and this is by TEM. And you can see the dimensions are about the same, but you get different information from the two techniques. AFM allows us to look at the shape of viral sized particles under ambient conditions. There's no prep other than to clean it and deposit it on a surface. There's no dehydration. There's no coating with metallic coating like you would with atomic, electron microscopy. These viruses are about 160 nanometers in diameter. Keep that number in mind. And this is just a cross-sectional diagram of what an enveloped virus like EHV163 looks like. We, after many trials and many issues, we were able to finally acquire virus Raman spectra from individual viruses or virions. The target is shown here. The arrow points to the target. The upper spectrum that you see here is from that single virus. The middle spectrum that you see here is from 5 individual viruses that have been averaged, and the brown shading around the mean, which is the black line, is the standard deviation of the measurements. And the lower spectrum was acquired from aggregates of the same virus in the same sample. And this is the average of 20 aggregates. Okay, so what do we see? We see signatures for DNA. These 2 peaks here are signatures for DNA. We see a peak that is signature for the amino acid phenylalanine, which is contained in almost every known protein. So this is a very useful diagnostic peak that we'll come back to in a little bit. We see peaks here that are attributable to lipids, which is diagnostic for the envelope that the virus has. So we were very excited to be able to produce a Raman spectrum from one single virus. And it tells us again that we've got DNA, we've got protein, we've got lipids. Not all targets that looked like viruses yielded the same information. And this puzzled us for a little bit. So we did a parallel TEM study, and we noticed in the TEM images that we would see many of these ghost-like electron transmissive objects that were exactly the same size as the virus shown here. So this is a complete virus. Do we have, are there any, if you have a question, why don't you just shout it out? Because I'm having trouble reading the chat. Okay, so we were seeing these empty, what we think are empty membranes, and sure enough, when we took Raman spectra of them, that's the upper spectrum here, we did not see any signature for DNA or a minimal signature for DNA. We didn't see a signature for phenylalanine, but we did see signatures for the lipids. And so our interpretation of this is they are empty envelopes. We don't know why they're empty, but they were in the sample, and we can recognize them from their Raman signature. Here's the complete virion again. And this is this bottom spectrum, or lack of spectrum, is just from an empty field on the substrate that we deposit the virus before we take the spectrum. And by the way, we deposit all of our cell samples and viral samples on mirrored finished stainless steel. So we're not using glass or quartz. We use slides that are made of stainless steel, which gives us really, really good results, and no background interference from the stainless steel, which is outstanding. Okay, so in one set of experiments, we raised E. Huxleyi in both light carbon and heavy carbon. So the spectrum, this is a spectrum from an individual E. Huxleyi cell that we acquired in 20 seconds, I believe. And it shows the diagnostic peak for phenylalanine. Remember, I mentioned that a couple slides ago. So that's the amino acid that is very sensitive to stable isotope enrichment. And this is a E. Huxleyi cell that has been grown in heavy C13 or C13 bicarbonate. And you'll notice that rather than a single peak at 1002 wave numbers, we have multiple peaks. So we'll come back to that in a second. Cells that were, viruses that were produced from these cultures after we infected them, we then took spectra of the viruses that were produced. Here is the viral spectrum from the isotopically light or natural abundance virus. Here is a Raman spectrum from viruses that came from isotopically heavy hosts. If we just look at the region around phenylalanine, and down here, this is natural abundance point. The fractional abundance is 0.01 or 1%. We see that there is just one major peak at 1002 wave numbers. And in the EHUX that has been grown for several days in isotopically heavy media, we get 4 peaks. We get peaks 966, 977, 988. The upper panel here in C is the virus, and the virus shows the exact same pattern. So we've demonstrated with Raman that as one would predict, the virus inherits the same isotopic signature as the host that produced them. That shouldn't be a surprise, but the ability to be able to demonstrate that and then document the level of labeling, which was within analytical uncertainty exactly the same, shown here, 65% versus 63, but notice the errors. So they essentially are statistically the same. This would allow us to do experiments with active and inactive hosts, isotopically light and heavy, and then look at the individual viruses released. At least that's the goal. Okay. So that's our viral story, and now the last vignette is looking at predator-prey interactions using Raman, and again a model system in culture in the lab. We've taken it to the field, but we haven't worked those results up yet. So the idea here is that we grow prey items, and in this case, it happens to be E. coli, just for ease of cultivation. We grow it in isotopically light media, isotopically heavy media, until they are uniformly labeled, till they come to stationary phase, basically. Once they have assimilated all the label that they're going to assimilate, we stop them from growing by subjecting them to UV light. This is a strain that has a UV sensitivity, and it shuts down growth. Doesn't kill the cells, doesn't stop metabolism, but they can't divide. We then clean up a ciliate host, protozoan, which is a lab rat for lots of protozoologists, and it's easy to manipulate in culture. And we do grazing experiments. So we introduce one set of incubations where it's isotopically light prey, one set of incubations isotopically heavy prey. And then through time, we measure grazing rates by disappearance of the prey we do cell counts, and we do Raman analysis. Just real quick, I don't want to dwell on this. It's basically to demonstrate that E. coli grown in heavy and light media grow exactly the same way. Those are overlapping growth curves from isotopically light and heavy. And when we grow it in the isotopically enriched media, an individual E. coli spectrum around the region of phenylalanine, again, that diagnostic peak looks like that. If we look at the time course just for the prey, so that's what's being shown here, at the beginning of the experiment, again, focusing on phenylalanine only, here's the entire spectrum, but most of that information we are not using. Through time, we see the appearance of peaks that are redshifted. We see through time the disappearance of the isotopically light. And at the end of an incubation, we see that almost all of the phenylalanine is heavy. We then surveyed the prey population by Raman and measured the fractional abundance of the cells, how much C13 per cell. We compared that to predictions based on growth curves that we made just by using old-fashioned optical density or just spectrophotometry. And so the blue curve is what was measured directly cell by cell using Raman. And the other two curves, whatever colors those are, gray and yellowish, are the predicted labeling rates. And you can see that they overlap perfectly. Now we do the grazing. We look at the grazing experiment and again, focus in on phenylalanine. And we look at it through time, and within an individual ciliate, they start out completely isotopically light. In a matter of less than a day, 19 hours, we see C13 gaining dominance, and then that trend continues all the way to the end, as one would expect. Okay, and if we look at the individual cells through time, we can do two-dimensional maps, not just measure one spot in a cell, not take a single spectrum, but take multiple spectra. We set up a grid, and I'll show this in more detail in a moment. If we look at it early in the time course, it's 6 hours, and the color coding is isotopic abundance. The warmer colors mean it's isotopically heavy. The blue or the cool colors mean isotopically light. And we can see regions of the predator cell, this is the tetrahymena, that at 6 hours are focal points for that heavy isotope. And in this case, these several hotspots here are food vacuoles, where it's basically the prey inside the vacuole rather than the biomass of the predator. They haven't been fully digested yet. But by 27 hours, a little more than a day, we see almost uniform distribution of that carbon-13. So you can imagine that we can do these experiments over finer timescales to really trace the locations of the stable isotope. To produce one of these maps takes about 3 hours. Each spectrum and each map has just under 400 spectra to produce it. Basically we're doing it at 100x magnification. Okay, we asked ourselves after this experiment, seeing that map in 2D, can we do similar things in 3 dimensions with cells that have never been dehydrated, never been filtered, just preserved? The only requirement is the cell can't be moving. It's got to stay still so we can produce the map. So my postdoc Felix Weber developed a technique where we actually put the stainless steel substrate underwater in a small petri dish. By pipette, he deposits a cell suspension of his ciliates and onto the slide, and we have a water immersion 63x objective lens. And basically we set up an experimental grid that looks like this figure right here, where we do multiple maps at 7 levels. So each level is shown here on the right. And we're showing maps of the distribution of protein and the distribution of lipids. Just as a demonstration, I suppose it's useful, but we weren't looking at isotopes in this experiment. We just wanted to know, can we look at macromolecular distribution in fully hydrated cells that have never been compressed, look at them in their lifelike state? So again, per plane, there were just under 500 spectra. At each node, we take a spectrum, and we do that in 7 planes. Okay, lovely. So we can look at this image from 3 different dimensions, and we can go a step further and actually produce, come on now, let's see. 3-dimensional image. Oh my goodness. I gotta, here we go. I'm sorry. This is animated. It's supposed to, it worked earlier. I don't know why it's not working. Sure. All right. So I'm not going to take up more of your time, but imagine this rotating around and around. We can look at that cell in 3 dimensions, fully hydrated, and map out whatever we care to map. In this particular image, we've mapped out the distribution of DNA in red, the distribution of protein in blue. And yeah, that's disappointing. Anyway, we can look at it in sections in any axis we want: longitudinal, sagittal, cross section, and look for distributions of lipids, isotopes in a SIP experiment, DNA, and so on. So what we're seeing is macronucleus, micronuclei, and perhaps even food vacuoles that have DNA. So we were very excited by that. And this is a cell that's 40 micrometers in diameter. Okay, believe it or not, I've come to my summary slide. So we're pretty enthusiastic, as you might imagine, about this approach. And some of the things to recommend Raman is that the requirements for sample prep are very relaxed. We can, as long as the cell is standing still, not moving, we could look at live cells. We can look at dried cells. We can look at preserved cells. We can look at frozen cells in a block of ice. We have a stage, a cryo stage that we can put on the microscope and look at things through ice. And it's ordinary lab conditions. We don't need high vacuum. We don't need sputter coating. We don't need any of that. From looking at shifts in diagnostic peaks, looking at shifts in wave numbers and diagnostic peaks, allows us to look at isotopic incorporation. In addition to carbon, we can look at deuterium, we can look at nitrogen, and we can also look at sulfur. And so we're calling it a proverbial single cell mass spectrometer. The techniques that we and other folks that are using Raman are enabling us to look at both intra and interpopulation variability in microbial processing of important elements in the ocean or in any ecosystem that you care to think of. SIP Raman fish is one of the few tools that can directly link function with identity. SIP nanosims also has the same capabilities. Raman microspectroscopy and AFM provide windows into this microcosmos, and that enables us to analyze variability of physiology on a single cell basis. It gives us tools to look at pathogen-host interactions, tools to look at microbial predator-prey relationships, symbiotic and parasitic exchanges. And we can do 2-dimensional and 3-dimensional mapping of individual cells, looking at target biomolecules as well as stable isotopes. And lastly, the only limitation seems to be our imagination, time, and money. And lastly, I'll leave you with some food for thought. In our single cell growth rate experiments that we published, we observed that in a clonal Synechococcus culture, clonal meaning came from a single parent cell, we saw that the difference in growth rates was about 27%. That's the coefficient of variation around the mean at any given time, at any given level of labeling. And that's under homogeneous, controlled laboratory conditions. So what does that suggest about the variability in a population's activity and growth in a natural, heterogeneous, microspatial seascape? I would predict that that variation would be much, much greater. And what does that suggest about interpopulation genomic and transcriptomic variability? We also imagine that that could be very, very high. And again, bulk techniques that we use are blind to such variations. Okay, I have concluded my talk. I thank you for your time and attention. This is my lab group here outside of my building. Hats off to CV Raman and the NSF and the Moore Foundation for their support. And now I'm ready to discuss. So please chime in.